In the last webcast, we touched on three fundamental classes of mechanistic problems: sequence of steps, site selectivity, and stereoselectivity. Now, our goal is to understand the experiments that will allow us to shed light on solutions to these problems. In the literature associated with your MOMADAC topic, you'll find many examples of the application of the scientific method to these fundamental problems. We'll discuss some of these methods in a general sense in this webcast. The essential idea is that a hypothesis is generated, then an experiment designed and executed to test that hypothesis. Hypotheses identify a specific origin for an effect, and the associated experiments vary that origin and look for a change in what we might call the response of the reaction, usually its rate or yield. Let's begin with the sequence of steps issue. The key step to address in a mechanistic sequence is the rate determining or slow step, which essentially dictates the speed of the overall reaction. To determine the nature of the rate determining step, we may alter the structure of the substrate or the reaction conditions and examine the effect on reaction rate or yield. For instance, consider this example from the last webcast. How will the polar and concerted mechanisms operate differently under different reaction conditions? Can we set up a situation in which one mechanism should produce a change in yield or rate, while the other one should not? The answer is yes. By altering the polarity of the solvent, we can determine which mechanism is most likely operating. If the polar mechanism is operating, the rate of the reaction should change substantially upon a change in solvent, since the polarity of the solvent affects the stability of the polar intermediates. On the other hand, changes in solvent should not affect the concerted mechanism, which lacks the buildup of charges. In practice, small solvent effects are observed, supporting the concerted mechanism. For enzyme-catalyzed reactions, the exact bonds and atoms involved in a mechanism may be unclear. The simplest way to identify the most likely mechanistic players is to assume that a particular amino acid participates, synthetically alter that amino acid within the enzyme, and see the effect of the alteration on reaction rate and yield. In one example of this so-called site-directed mutagenesis approach, mutation of a lysine residue in an aldolase enzyme led to the complete loss of enzyme activity, demonstrating that a reaction between lysine and the substrate is essential for catalytic activity. Let's move on now to the origin of site selectivity. In enzyme-catalyzed reactions, site selectivity may be controlled by amino acid side chains that don't participate in the reaction directly, but help to bind the substrate in a particular conformation. Using site-directed mutagenesis or other techniques, we can alter these amino acids and observe the resulting effect on site selectivity. In other words, we can look and see if any other isomers form. An intriguing example of this idea comes from the field of enzyme-mediated DNA modification. Altering the primary sequences of DNA-modifying enzymes results in a change in the selectivity of the enzymes for a particular sequence of nucleotides within DNA. Site selectivity in laboratory reactions generally arises from either electronic or steric factors. By steric factors, we just mean the spatial accessibility of each functional group to an incoming reagent. By electronic factors, we're referring to whether each functional group is under the influence of an electron donating or withdrawing group nearby. To determine which set of factors is at play, we can examine the behavior of a series of substrates in which sterics or electronics are systematically varied. For instance, the reaction you see here exhibits site selectivity for one of two double bonds. Since the steric properties of the two alkenes are roughly the same in this substrate, we establish the hypothesis that electronic factors are at play, then run the reaction with a simple series of electron-poor, neutral, and electron-rich alkenes. Sure enough, the electron-rich alkene reacts most rapidly, showing us that the difference in electron density of the two double bonds is the source of site selectivity in this reaction. The origins of stereoselectivity can sometimes be difficult to pinpoint, but for enzyme-catalyzed reactions, crystal structures or computational models can often shed light. Our fundamental hypothesis here is that the chiral environment of the enzyme preferentially exposes one of two inequivalent functional groups or faces in the substrate to an incoming reactant or amino acid. Experimentally, what we can do is crystallize the protein with the substrate or an analog as the ligand, then examine the arrangement of bonds and atoms within the active site. Openness of a particular functional group or face over any alternatives provides evidence of the origin of stereoselectivity. Analyses for laboratory reactions are similar. However, since reactive intermediates cannot be observed directly or crystallized like enzymes, computational studies and rationalization are more common. The past 2 webcasts have armed you with a general framework for thinking about problems, hypotheses, and experiments associated with organic reaction mechanisms. In the second half of this lesson, we'll learn how to communicate these ideas for your specific topic in the form of mechanism challenges.